Hi, I'm Shil and my partner is Brian, and we're representing Group 13. The article that we will be discussing today is titled Requirement of DDX DEADBOX RNA Helicase for HIV Rev RRE Export Function. Just to preface things, DDX3 is the DEADBOX helicase. The virally encoded Rev protein binds a highly secondary structured element called Rev response element, RRE for short, which is present in all unspliced and partially spliced HIV transcripts. Rev binds to RRE and directs HIV transcripts from nucleus into the cytoplasm. Rev interacts with chromosome maintenance region 1, CRM1 for short, which is an export protein. This brings us to our central question. What is the function and mechanism of DDX3 in HIV1 export and the Rev RRE CRM1 pathway? The hypothesis is that human DDX3 serves like yeast DBP5P in restructuring cargo RNAs to permit their translocation across and dissociation from the nuclear pore. To begin to understand DDX3, the researchers analyzed the sequence of DDX3. It contains all 8 motifs conserved by eukaryotic RNA helicases, as you see in the highlighted red boxes in Figure 1A. Investigation was then done into whether DDX3 was an ATPase and if it was also a helicase. To do this, they made 2 DDX3 point mutants. One was formed by changing lysine into glutamic acid at position 230 in ATPase motif. This was called DDX3 mutant 1. A second one was formed by changing serine into leucine at position 382 in the helicase motif. This was called DDX3 mutant 2. A negative control in wild-type DDX3 was also added, and then ATPase activity was tested by hydrolysis of ATP in the presence of RNA. Results show that DDX3 hydrolyzed ATP efficiently, as seen in Figure 1B, lane 2. However, DDX3 mutant 1 lost ATPase activity, while DDX3 mutant 2 had its ATPase activity significantly reduced, as seen in Figure 1B, lanes 3 and 4. Next, the researchers tested DDX3's ability to function as a helicase. The wild type and 2 mutants were incubated with double-stranded RNA. Results show that DDX3 unwound RNA duplex in an ATP-dependent manner, as seen in Figure 1C, lane 2, but was inhibited by competing amounts of non-hydrolyzable gamma-thio ATP in Figure 1C, lane 5. Both of the point mutations eliminated DDX3's RNA unwinding activity, as seen in Figure 1C, lanes 3 and 4. This shows that DDX3 is an energy-dependent RNA helicase.
Main idea is DDX3 enhances the Rev protein, Rev responsive element dependent expression. The Rev responsive RRE containing plasmid is shown at the top of Figure A. There are 2 bar graphs measuring the increase in cat protein activity from the cat reporter gene in 2 different cells. There are 10 lanes in total, each with its respective control vector or DDX3 variant. Whether the control helicase or Rev are present is denoted by plus or minus sign and a microgram amount if applicable. When overexpressed in either cells, DDX3 didn't affect basal expression from the plasmid, as seen in lanes 2, 3, and 8. However, co-expression of Rev protein with DDX3 allowed Rev-dependent plasmid expression to increase, as seen in lanes 5, 6, and 10. For Figure 2B, the new plasmid encodes GAG protein, which is a group-specific antigen and is a structural protein of HIV1. GAG expression is highly responsive for Rev, as seen in the gels and bar graphs. Rev-dependent expression of both P24 and P55 GAG were enhanced by co-expression of DDX3. At the bottom is the beta-actin control, which should appear.
The unspliced and partially spliced versions of HIV1 contain RRE and are Rev-dependent. PNO43 is a molecular clone of HIV1. This was used to co-transfect cells with either DDX3 or its antisense version, DDX3AS. Differences in cytoplasmic versus whole cell RNAs were analyzed using Northern blotting, and no differences were found, as seen in Figure 4A, lanes 3 and 4, and the table quantifying this below it. Also, the Rev RRE independent 1.8 kilobase transcripts were similar throughout all 4 lanes. However, in lanes 1 and 2, you see a 5-fold reduction when using DDX3AS versus the wild type, as this shows that DDX3 is exporting HIV1 into the cytoplasm. After this, the researchers tested whether DDX3 shuttles between the cytoplasm and the nucleus. HeLa cells were transfected with HA-tagged DDX3 were treated without leptomycin B, which is an inhibitor of CRM1 mediated nuclear export. Without LMB, DDX3 was prominently cytoplasmic, as you can see in figure 4B with no LMB. The fluorescent parts are on the outsides of the cells and not the nucleus centers. However, you add LMB in any quantity, the DDX3 is now located in the nucleus. This shows that DDX3 is a nucleus to cytoplasmic shuttling moiety. Next, this was tested to see how exactly DDX3 binds to CRM1. Co-precipitation of DDX3 with CRM1 was tested. Figures 4C lanes 2 and 4 show that these do co-precipitate. Figure 4D verified the association of DDX3 and CRM1 by also testing phosphodiesterase Na and murine cycline T1. Immunoprecipitation showed that DDX3 physically bound only to CRM1 as seen in figure 4D lane 2. Finally, DDX3 complexing with REV was tested using GFP and GFP-REV. Immunoprecipitation brought down GFP-REV, but not GFP alone, as seen in figure 4E, lanes 3 and 4. This shows evidence leaning towards there being a CRM1 DDX3 REV ternary complex.
The key point is DDX3 co-locates with nucleoporins at the outer nuclear rim. Figure 6 shows various Western blots. The plus or minus signs at the bottom determine whether HA tag DDX is present. For the top panel, lysates are probed with anti-HA and nothing communoprecipitates for the first 2 lanes, while the last 2 lanes have communoprecipitations. The middle panel shows immunoprecipitation of nucleoporin 62 by anti-nucleoporin 62. The isotype controls have no bands because they lack specifics, target binding. In the bottom panel, communopprecipitation of HA tag DDX3, which is lane 3, occurs with a nucleoporin containing complex. When GFP DDX3 was visualized, co-localization of DDX3 as a rim with the nucleopores occurred. GFP Sun1 is the constructed GFP tagged internuclear membrane protein Triton X. The detergent removed the outer nuclear membrane but left the internuclear membrane intact, and the signal for inner nuclear membrane protein Sun1 was unchanged as seen in panel 16, while the signal for GFP DDX3 was removed as seen in panel 7. Therefore, DDX3 was on the inner side of the nuclear membrane and is shuttled through the pore to the outer side. The last key point is DDX3 limits HIV-1 replication. In figure 1, cells were transfected with HIV-1 molecular clone BH10 and either DDX3 or DDX3 mutation 1, and the culture supernatant for P24 GAG production was analyzed at 3 time points compared to the vector control. Wild-type DDX3 enhanced, while DDX3 mutation 1 suppressed P24 production. Chimeric HIV-1 genomes which contain DDX3, DDX3 mutation 1, or a non-expressible DDX3 due to a start codon being replaced by 2 consecutive stop codons at the NEF region for plasmid NL43. Based on the P24 profiles on figure 7B, plasmid containing NL-DDX3 replicated better than the other 2 in both cells. Figure 7C compared plasmid containing NL-DDX3, which is the plasmid containing the chimeric HIV-1 genome, to the wild-type PNL43, which is the HIV molecular clone. And the graph on 7C shows that despite the JORCAT cells having high endogenous levels of DDX3, chimeric plasmid replicated better. In conclusion, although DDX3 was induced by TAT, it plays no role in HIV-1 transcription. Instead, DDX3 is an RNA-dependent ATPase and helicase which functions in the REV RRE CRM1 pathway for the export of unspliced and partially spliced HIV-1 transcripts. We show that DDX3 is a nuclear cytoplasmic shuttling protein that binds CRM1 and decorates nuclear pores, and whose function depends on its enzymatic activity. One outstanding question is the involvement of additional cellular participation in these export paths isn't completely understood, and here are our sources.